Thank you for opportunity to present, let me present our work on antibiotics which are involved in treatment of acne. So acne is the most common skin disease, as you know, and deep understanding of the molecular mechanism regulating pathophysiology and inflammation in acne is needed to determine crucial targets for drug design and to develop comprehensive treatment strategies. Tetracyclines are the most frequently prescribed antibiotics by dermatologists, and they are known for their antibacterial and anti-inflammatory effects. There are 3 generations of tetracyclines, and you can see that the 3rd generation took a long time to come to the market. Today, we will focus on sarecycline. It has modification in C7 position because it's a first known tetracycline which has a narrow spectrum activity. When we use drugs which are broad spectrum, it can be a lot of negative side effects. Even short term of using some antibiotics can take up to 2 years for microbiome to come back to the original state. Also, using of broad-spectrum antibiotics may create a lot of side effects like gut dysbiosis, dysregulated metabolism, and what we will talk today a little bit, creating antimicrobial resistance. C. acne develop resistance to many antibiotics, and this is the data worldwide for doxycycline, tetracycline, and minocycline, and it's very warning that resistance is increasing. If you look at the local data, you can see that resistance to doxy and tetracycline in Jordan can be more than 30%. So sarecycline can replace doxycycline and minocycline as a first-line narrow-spectrum antibiotics treatment in order to reduce antibiotic resistance, maintain human microbiome homeostasis, and decrease risk of adverse effects. So we need to know how this antibiotic works. This is the target for all tetracyclines. It's bacterial ribosome or 70S particle. It has a large subunit, 50S, and a small subunit, 30S. And if we will slice it in half, you can see inside the ribosome there is an AP and E site tRNAs. tRNA deliver aminoacyl moiety to the ribosome as well as established codon-anticodon interaction with mRNA. That's a decoding center. And then at P site, there is a peptide bond formed, and the nascent peptide will go through the tunnel out of the ribosome. And E site is where they exit. This illustrates how peptide synthesis is working. So tRNA delivered to the A site, then peptide bond is formed, then its translocation together with its peptide goes in the tunnel, and that's everything dissociate and start again. What's important here is that common tetracycline binding site is in A site of the ribosome. How we do our studies? We use X-ray crystallography to look at the molecular structure of the molecules. So in our case, the crystal of the 70S ribosomal subunit will expose to the X-ray beam that will create a diffraction pattern from which we calculate electron density map in which we can build molecular model. So we now focused on the structure of the ribosome with sarecycline and ribosome purified from the model organism, Thermus thermophilus. So this will illustrate one of the mechanisms of resistance to antibiotics. This is a 70S ribosome, and tetracycline is in red. It binds to the A site of the ribosome. In blue is a tRNA in the P site. In magenta, it's a mRNA. So this is a tetracycline, and this is a model. There is a protein, which is a ribosomal protecting protein. It binds to the A site of the small ribosomal subunit, and its binding is overlapped with that of the tetracycline. That allow this protein TET-M to displace tetracycline from its site, and that restore protein synthesis. Now we have experimental data. It's our data of a serocycline bound to the same thermophilus ribosome. Serocycline is in red. Okay, sorry for this. So serocycline is in yellow here, and it's also bound to the canonical tetracycline's binding site. Our structure was done at the atomic resolution, so we can see all hydrogen bonding interaction of the serocycline with ribosomal 16S RNA. Our data shows that this C7 extension of serocycline is long enough that in case of the bulky nucleotides on the mRNA in the A site, it can establish new hydrogen bond which increase its binding compared to other tetracyclines. That may explain the increased potency of this antibiotic. It also explains how serocycline can interfere with resistance mechanism of bacteria, because that extension also long enough to block some binding nucleotides for this TET-M protein, the ribosome protection protein. So that's how serocycline avoid, in part, resistance to antibiotics. That is not happened for the regular tetracycline. Overall, this C7 position modification results, as I mentioned, to this overcoming ribosomal protection proteins, also efflux pump, but still some mechanism of resistance exists, like permeability for the drugs, inactivating of the drugs, and also mutation in the DNA to change the target. Serocycline also more active on C. acne protein synthesis, which is why it's a narrow spectrum activity antibiotic. To understand why is that, we decide to go for its clinical target, ribosome purified from C. acne. Because C. acne is a gram-positive bacteria, it's a part of the normal flora in the skin and play important role in pathogenesis of acne. Is interaction of serocycline with a model bacteria the same as with this clinical target ribosome from C. acne? To answer that question, now we use cryo-electron microscopy. That technique doesn't require have a crystal of the ribosome, so we don't need a lot of material, and ribosome put on the grid, freeze or vitrified, and then exposed to the electron beam, and from this we will have 2D pictures which we can reconstruct in the 3D cryo-electron map and build atomic structure of the ribosome with bound antibiotic. What we found when we look at C. acne ribosome, that the same site, classical tetracycline binding site, exists, but in addition to it, we found the new second site for serocycline, which binds in the peptide exit channel of the C. acne large 50S ribosomal subunit close to the peptidyl transferase center. This is a closer-up view. You can see serocycline binds very close to the known macrolide class of antibiotics. In this case, it's erythromycin. Also, if we superpose the structure of ribosome with known peptide in the peptide exit channel, we can see that it clashes with the serocycline in there, meaning that serocycline in this second site works as a plug, which doesn't allow protein to go through the tunnel. If we go back to the classical or canonical binding site, we found also some differences. There is no magnesium which mediates interaction of serocycline with the G950 nucleotide of the 16S ribosomal RNA, unlike in this model organism, Thermophilus ribosome, there is a magnesium. So this interaction looks like much stronger compared to this. So overall, interaction of hydrogen bonding of the tetracycline on the clinical target ribosome is a little bit different from what we saw on the model organism's ribosome. That also promote us to look at the interaction of the minocycline and doxycycline, second generation antibiotics, with the ribosomes of this model organism, E. coli, and also from the Cutibacterium acne. We use cryo-electron microscopy to look at these interactions, and we found that doxycycline, minocycline, and tetracycline revealed species-specific differences affecting drug interaction and occupancy. So now we analyzing all this interaction deeper, and this data will be ready for publication soon. In conclusion, our results redefine the mechanism of action for tetracyclines and provide a structural basis for the rational design of narrow-spectrum tetracyclines to overcome the rising threat of antibiotic resistance. The work was done in Christopher Bunick lab at Yale School of Medicine. Swapnil Devakart contribute a lot to this project. We also thank our collaborators from Yale, and these are our funding agencies, and special thanks to American Acne and Rosacea Society.